Good morning, my fellow scientists. It is Friday, May 5th, 2017. Happy Cinco de Mayo. Let's talk about science. I've been working on DNA nanotechnology lately, which is a bit of a far cry from my usual rants about batteries and iron and so forth, but I thought it would be interesting to talk a little bit about why I'm interested in that. There's a lot of good reasons to be interested in nanotechnology. When I was a young man, I was very interested in post-scarcity, building nanotechnology in order to eliminate the price of physical goods. I was really inspired by books like The Engines of Creation by Drexler and by The Diamond Age by Neil Stephenson. And I just thought, if I could contribute to anything, it would be the ability to assemble matter into arbitrary objects based on code. And we're a far distance from that. I kind of thought in 1999, when I graduated from high school, that I would live to see post-scarcity nanotechnology matter assemblers, and that would happen reasonably soon. In retrospect, that was a bit naive, to say the least. We're still a long way from that. But some things have happened in the last 17 years that have given me some hope. So one of the things that really made me think that nanotechnology, like they have in science fiction, was possible was DNA nanotechnology. So most of the time we think of DNA as being heredity, something that life uses to pass on instructions about how to build more life. And that's true, but DNA is also a chemical. It's just another chemical. Just a thing we can synthesize, a thing we can create. When you synthesize DNA, it doesn't correspond to a gene or an organism. It's just a string of monomers, the adenosine, cytosine, guanine, and thymidine monomers that can be in any order that we want. So when I say I'm going to make some DNA that has A, T, G, C, T, G, that's not from a critter. That's just a thing I can synthesize. And if I take that and combine it with its reverse complement, single-strand DNA, those will come together and make the double-stranded double helix that we're all so familiar with. That simple rule set that drives DNA assembly, that A binds T and G binds C, that rule set can be used to construct some pretty sophisticated objects. So people have taken virus DNA and folded it up into lots of configurations. They call it DNA origami. And in my imagination, that's about the closest thing we have to molecular nanotechnology, the ability to put matter into a configuration prescribed at the molecular level, but spanning many nanometers in size. That's just, in and of itself, that's just amazing. I mean, what applications that can be put to is still an open question. DNA is not diamond. DNA is not steel. It's not immediately obvious what you can manufacture out of DNA, but DNA does interact with biology in some interesting ways. So my hope is that these kinds of powerful assembly ideas can be used to build things that interact with biology. And I think we're getting closer to that, not least with aptamers. So aptamers are bits of DNA that have been evolved in the lab to bind to particular things. So if you want to, say, have a piece of DNA that binds to uranium, you can do that. You can evolve single-strand DNA to bind to uranium ions. That seems kind of odd. There's nothing in biology that likes uranium ions. And yet, DNA is capable of undergoing evolution to bind whatever. So what could we do with that? We could use aptamers to drive assembly, to drive readout, to do detection. We could do detection to drive assembly in some sort of computational format in order to make larger structures. I think that at this point, the technology is evolving so quickly that that's like asking, well, what can you use a computer for from the perspective of 1960? The kinds of things we can use it for will have to evolve as the technology evolves. And I'm excited to see that. So if you're interested in that kind of thing, I hope you'll tune in on Monday when we talk more about DNA and DNA nanotechnology and the chemistry of nanotechnology here in the Allen Lab.